Hello guys, you're welcome to my YouTube channel. Please endeavor to like, share, subscribe and comment. It is very important. My name is Tutor Joseph. Alright, so this morning I'll be taking us biology. This morning. Seed. Now what is the meaning of seed? Alright, seed means a ripened, fertilized and developed ovule. Alright, seed means what? A ripened, fertilized and developed ovule. Okay, please do not take it for fruits. Fruits is a ripened, fertilized and developed seed. Okay, ovary. Sorry. Now, a ripened, fertilized and developed seed, developed ovule is called what? Seed. A ripened, fertilized and developed ovule is called what? Seed. Fruit is a ripened, fertilized and developed ovary. Now, seed has different structures. Seed has like structures of the seed. Structures of the seed. Alright, definitely one, you have the seed coat or tester. Alright, you have the seed coat or tester, and two, you have the helium. Three, you have the micropyle. And four, you have the embryo. Alright, the seed has so many structures, and these are the seed coat or tester, helium, micropyle, and what? Embryo. Now, what do you understand by the word coat? Coat is just like a structure, is a sheet that surrounds or that protects a thing. Now, the seed coat or tester means the outermost covering of the seed. Alright, it is the outermost covering of the seed, and it houses the inner embryo. Okay, which means that the structure that houses the inner embryo is called what? The seed coat or the tester. Alright, in some seeds, the seed coat is made up of external tester, that is outer tester, and inner tegment. Alright, in some seeds, alright, the seed coat is made up of an outer tester and inner what? Tegment. Okay, now let me put this question on the board. A thin, transparent and membranous structure that surrounds the seed is termed A, tester, B, tegment, C, helium, D, micropyle. Okay, now the thin, transparent and membranous structure that surrounds the seed is termed tester. Please, the tegment I told you is the inner structure. Then we'll come to this now. Now, what is the function of the helium? All right, the helium, what it does is, okay, let me bring it this way, please. Let me bring it this way. Now the helium of a seed, all right, attaches the seed to attaches the seed to the seed stalk, and this seed stalk is called the funicle. All right, that is the work of the helium. The helium attaches the seed to the seed stalk, called what? The funicle. Now the macropyle of a seed, the macropyle of a seed means the tiny hole that permits the entering of air, water, and other required what? Nutrients. All right, for the seed. All right, so I said the helium means the part that attaches the seed to the seed stalk, called the funicle. Then the macropyle is that small hole that permits the entering of air, water, and other required what? Nutrients. So please, that is the work of the macropyle. Of course, you now have, all right, you now have the embryo. Now, definitely the embryo of a seed, is made up of 3 parts. The embryo of a seed is made up of 3 parts. You have 1, the plumule. This plumule is otherwise called the embryonic shoots. All right, shoots. Then 2, the radical, otherwise called the embryonic roots. And lastly, you have 1 or 2 cotyledons. 1 or 2 cotyledons, otherwise called the seed leaves. All right, so I said the embryo is made up of 3 parts: the plumule, the radical, and 1 or 2 cotyledons, otherwise called what? The seed leaves. Okay. Now this it could be monocotyledon or 1 cotyledon. Don't worry. After now we are going to be discussing germination. We now have hypogeal and epigeal. We now have monocotyledons to the hypogeal, while the dicotyledons to the epigeal. So that is how it is. But today we are discussing what? Seed dormancy, and how seed dormancy can be what? Prevented. Now, the plumule of a seed is protected by a sheath called, the sheath that protects the embryonic shoot is called the coleoptile. It's called the coleoptile, right? The sheath that is found covering or protecting the plumule or the embryonic shoot is called what? The coleoptile. Then the one found covering the embryonic roots, otherwise called the radicle, is called the coleorhiza. Alright? The coleorhiza is that sheath, that outermost covering that protects the radicle. Like I said we have 1 or 2 cotyledons. It could be monocotyledons or dicotyledons. So after now we'll be discussing that. Now, I haven't given you all these things you have here. Now let us now proceed to seed dormancy. Now, what is the meaning of seed dormancy? Seed dormancy. What does it mean? Seed dormancy. Now, the word dormant, to be dormant is like to be stagnant. Alright? That is to be in a state of a sluggishness, delay, like things are not just working well. Okay. Now, seed dormancy means the word seed dormancy. It means an inactive period of the seed. All right, it's the inactive period of a seed to germinate, and as such, it results to slow growth. It results to what? Slow growth. And 2, the seed eventually ceases to grow. Yes, it stops eventually because since there are factors, there are some external factors that contribute to seed dormancy, okay? And as such, the seed will at first might even try to spring up, to sprout. But as time goes on, it dies off, alright? It stops growing due to some external and what? Internal factors, okay? Now, so there are different ways in which seed dormancy can be what? Prevented. There are different ways in which seed dormancy can be what? Prevented. Alright, let's write it here. Ways to prevent seed dormancy. Now, in what ways can we actually prevent seed dormancy? Alright, now, number one, we can prevent seed dormancy by removing the tester. Removal of what? Tester. Through scarification. Now, what is the meaning of the word scarification? Of course, most of you or some of you might be asking yourselves, what is the meaning of that word scarification? Don't be scared, okay? I will explain. Now, the act of weakening or opening the tester of a seed through thermal means, chemical means, and mechanical means is called what? Scarification. I'll recap. The act of opening or weakening the tester by removing it through chemical means, thermal means, and mechanical means is called what? Scarification. Okay, that is for that. Then number two is exposure to fire. Exposure to fire. We are not saying you should burn it. Yes, we are not saying you should burn it. Don't go and just set fire, buy like 20 liters of petrol, put it, you set it ablaze. You now say, no, you are burning it. It's not that. We are not talking of that right now. Okay, exposure to fire. All right. 3, adding dormancy, breaking hormone. All right. Number 4. Now, another way to prevent seed dormancy is exposure to variable temperature, alternating temperature, low or high temperature. That is exposure to alternating temperature, alternating high and low temperature. Temperature is between 77 degrees Celsius and 100 degrees Celsius. So please take note of that. Now, the 5th step that is used to prevent seed dormancy is treatment with water or digestive enzyme. Alright, so these are the various ways in which seed dormancy can be prevented. I said seed dormancy means an inactive period in which the seed refuses to grow effectively, and as such it grows slowly. Then it gets to a point it ceases to grow and eventually gives up, dies off. Alright, and it's as a result of certain factors. Okay, and I said that the seed has different structures. The helium, which attaches the seed to the stick stalk, called what? The funicle. And that, I said it starts a macropyle. The macropyle permits the entering of air, water, and other required what? Nutrients. Okay, and Yes, the seed also requires oxygen. Due to the fact that the seed is an organ, all right, it requires oxygen for what? Respiration, in order to produce energy in the form of what? ATP, adenosine triphosphate. All right, so at this point, to call it a day on this topic, seed/seed dormancy. Okay, now in our next class is going to be germination. All right, what is germination, the types of germination, and the characteristics of diverse types of what? Germination. All right, let me do a brief introduction. You know, germination is a process that involves the gradual development of the embryo of the seed into seedling or young plants. All right, as soon as a given seed is planted, the embryo grows out from it and forms a new plant. All right, it obeys, or it goes with certain mechanism. It's like a thing that occurs in a physically, like physics by physics. All right, so take note of that. Please endeavor to share, like, comment, and subscribe. Please, it is very, very important. Thanks for watching. Remember, my name is Dr. Joseph, and the name of the tutorial is the explicit what? Tutorials. Please make sure S is attached. All right, you can call me on 091 6011 5375. You can WhatsApp me on 4418387. Do have a wonderful day. Thanks for watching. Thanks for choosing the explicit tutorials.